Welcome, everybody. We're going to be moving on from Chapter 1, Theories, Theorists, and Research Design in Human Development. We will be revisiting those as we go through the semester for each particular stage of development. But right now, we're going to move on to Chapter 2, and this is Biological Foundations, Heredity, Prenatal Development, and Birth. We'll be learning about genes, DNA, how that affects our behavior, and then we're going to go on and learn about preconception all the way to the birthing process during this chapter. I added some supplemental videos for you to watch in the same section as you're watching this video, and it'll be on actual genes, DNA, alleles. Also, there'll be a video on what PGD is, a video on spinal bifida, and there'll be a video on preconception to birth. So that video is very good. I want everybody to watch that one, and you'll understand the whole process of that prenatal development. So we're going to go on to the next slide, and we'll start talking about genes. So the period from conception to birth is the foundation for all of life's development. Our objectives with Chapter 2, we're going to be talking about how do parents contribute their genetic material to their child? What are the stages of prenatal development? What are some risk factors that can affect pregnant mothers and their unborn children? And why do some births have complications while others do not? So the first series of our lectures for Chapter 2 will begin with genetics. 2.1. In the beginning, there were 23 pairs of chromosomes. So our learning objectives for this part of the chapter are: what are chromosomes and genes? How do they carry hereditary information? How is children's heredity influenced by their environment? So let's begin with the mechanisms of heredity. So when we think of heredity, we think of what do you get from your mom and what do you get from your dad? And how does that make a human, or in your case, you? So what we're going to look at first is the basic part of heredity, is chromosomes. And that's the structures that contain all our genetic material. And you get some from your mom, and you get some from your dad. And then combine them together makes you. So each person has 23 pairs of chromosomes. 22 are autosomes. And the 23rd is the sex chromosome. And that's what makes you a boy or a girl. So XY is a boy, and XX is a girl. Deoxyribonucleic acid, or fancy word for DNA, is the molecules that composes one chromosome and is the biochemical basis of heredity. So basically you have a DNA, you have your cells in your body, and you have your DNA as your blueprint. The blueprint tells the cell what it's supposed to be. So you have cells for your eyes, and you have DNA pattern for your nose and how tall you're going to be. So each cell has their own DNA pattern in it. So we're going to go through and look how these chromosomes become you and become me. So now let's take a look at it in a functioning way. So remember, you have your 23 chromosomes. You got one part is from your mom and one part is from your dad. And as you can see on the left-hand side, that you see they're numbered 1 through 23, and you can see the last two are your sex chromosomes, so they look a little different. And those all together are called, one of those inside your 23 is called a human karyotype. And a karyotype is an ordered display of one individual's 23 pairs of chromosomes. So that's the representation of all 23. So each person has a unique blueprint. That is your blueprint on how you are made. So what does that mean? So how do you get started from there? So you have your chromosomes, and we're going to learn this as we go a little bit later in the preconception, but you're going to start with your cell. You're going to start with that chromosome. You're going to get one from your mom, one from your dad, and each cell, so you can see in that picture, there's the cell and the nucleus. Each cell has a chromosome inside the nucleus. So most DNA is in the nucleus, and that's going to tell the cell what it needs to become. So you need to think of your DNA as the blueprint to your body. So every cell in your body has the DNA in it, and it's going to tell it what's to be, so that you don't have 50 livers in your body or 5 eyeballs. It's going to say, I need you to make 2 eyeballs. These cells are going to make eyeballs. And then you have cells that say, I'm going to make livers. And you have cells that say, this is skin. So all those cells and those, as you can see, those chromosomes have specific markers on them that are going to tell the cell what it needs to turn into. Now, as we go across this picture, you're going to see how that, if we break down that chromosome even further, what is inside it? Well, DNA consists of a long strands of paired nucleotides, and it's arranged in a double helix. So most people, we've seen this double helix, may not have understood it, but we've seen it. So basically what that means is your DNA, that's your blueprint that's going to tell what the body what to make. Well, how does it know what to make? What's the code? So all those letters in there is the code. So your DNA is the recipe book, and then you're going to open up, and the genes are the codes or the recipes on how to make everything. So you're going to have a recipe or a gene code on how to make a liver. And how does that say? So the genes are going to be formed into patterns, just like you would form words into patterns in a recipe book. And it's going to say, if I have, as you can see, there's A, T, G, C. Those are those nucleotides. That's what makes up a gene. And so those different proteins and those different nucleotides, they make up a gene, and that what build on each other to make up the organ that you're going to make. So if I'm going to have a recipe or a gene code for eyes, it may be AT, GC, AT, AT, as you can see in there. I'm just making that up, but that could be the recipe for an eyeball. And then another recipe, if I want to make a liver, would be maybe TA, GC, AT. So each organ or each part of your body has a specific gene code that tells it what to do. And just remember, a gene is a series of those paired nucleotides, which are the A, T, G, C, at a certain location on a chromosome that codes for a certain protein. So remember, just think of it like this. You've got your book. Your DNA is your recipe book. Your genes are the recipes. And then those gene codes are the words. So everything in your body has a specific recipe on how to make it. And the gene codes are just the words on telling the body how to make it. So that we become who we are. So we're going to look a little bit more in the next slide and go over a little bit more on chromosomes, DNA, and genes. So let's review the genetic foundation. So this isn't a genetics class, but we need to understand this so we can understand are the genetic factors in development. So let's go over it one more time. So we have chromosomes. So remember, those are those rod-like structures. Remember, it's that X. It's that rod-like structure, the wound up DNA within the cell. Remember, most chromosomes DNA is in the middle of the nucleus, and it stores and transmits genetic information. So it's the blueprint of who you are. So DNA is the chemical substance that makes up the chromosomes. So chromosomes is the whole structure, it's the whole X, and the DNA is all that strand, that double helix that makes up the chromosome. And the gene is the segment of the DNA along the length of the chromosome. And the gene is remember, it's the words in your recipe book. It's the specific codes or recipe to make each individual organ. So the chromosome is the whole recipe book, and then you open it up, and the DNA is all the pages in the book because it has everything that you have, and then the genes are the actual written words to say what to do. So if you think of it like that, chromosomes is the book, is the recipe book, DNA is the recipes, it's the blueprint, and the genes are the words on there to tell every cell in the body what it needs to turn into. So now we're going to look at that DNA, that double helix, in a little bit more detail. So remember, DNA is the blueprint for what makes up your body. It's the instructions to tell the cells on how each cell needs to act, what it needs to turn into. And then the genes inside there, which are those nucleotides. So we see those GCTA. Those are the specific recipes on how to turn into it, what it needs to turn into. So their DNA is organized in the double helix with strands of phosphates and sugars linked by chemical compounds. And that's what makes up a gene. You can see those strands of phosphates and sugars is the ribbon part. And then the nucleotide bases are those 4 letters we were talking about. And as the way that those letters are combined makes is the recipe for making your eyeball. So if a recipe for making an eyeball might be ATGC, GCTA, and you combine those in 50 different ways, and all that combination makes up a certain structure of the DNA, and that tells that cell that's an eyeball. They understand that. So what we need to understand is that nucleotide bases can only be paired with each other with 2 different letters. So an A has to go with a T, and a T have to go with an A. So they either can go AT or TA. And the same thing is with G and C. So it's either G to C or C to G. And then the order of that A, G, C, T is what makes up our specific genes. And remember, the genes are the recipes. It's what tells the cell what it needs to turn into. And those nucleotide bases are the words put together to give those instructions. So let's just review gene, and then we're going to kind of group the genes into 2 different types. So I know I'm going over this a lot, but it's really important to understand so that you don't get lost in all the confusion of it. So remember, gene is the biochemical instructions, it's the recipe that is produced by a group of chemical compounds, so that's that ACGT, within a strand of DNA. Humans share approximately 99% of their genes with each other. So we have 99% of the same genes. Just the way that they're put together is different. That's why we all look different. Our recipes are put together differently, but we have all the same ingredients, okay? So we just look a little bit different, but we can all tell that we're humans. So then it's broken down into subgroups of genes. So you have a genotype and a phenotype. A genotype is a person's complete set of genes. So that is the total amount of genes. It's the genetic instruction, and so that is all your genes. That's your genotype. A phenotype is observable features that result from the interaction between a person's genes and the environment. So you might be asking, what's the difference? So a genotype is your complete set of genes that you were born with. You have them, you were born with them, that's your DNA. The phenotype are actually groups, they change depending on the interaction that genes have with the environment. So genes are your blueprint, and the environment can influence how they're put together. So, you have a blueprint, like you're going to put a house together, but you have the contractor that comes in and interprets it. Well, this is a bathroom, but I'm going to put, extra tile here, and I'm going to put an extra shower there. It's my environment. I'm going to shape it. It's still a bathroom, but it's going to look a lot different than maybe your bathroom. So that's the phenotype. We're still humans, but we're going to interact differently because of that phenotype. The environmental factors that has influenced your genes are going to result in them acting differently than, say, your friend or even your sibling. So we're going to go into this a little bit more in depth. So remember, we have 46 chromosomes, and you have 20,500 genes. So that's a lot of genes, right? So it's a lot of combinations that makes us so unique and different. So remember that a genotype is an individual's genetic information. So that is what you were imprinted when you were born, when you came out of the womb. A phenotype is the individual's directly observable characteristics. So it's how the environment shapes those genes. We are all humans. We are all born to walk, talk, breathe. But how does the environment shape those genes to make the person that we are? Just the characteristics. Do we, are we outgoing? Are we shy? Can we, do we talk a lot? All those types of things. We're going to go over that a little bit more in detail, But just think genotype is your individual, it's your genetic blueprint. It's what you were given at birth. And phenotype is going to be the characteristics. It's going to be how we shape that, how the environment molds that blueprint into who you are today. Because you could take that same person, think of twins, and you separate them. You put them in two different environments. They have the same genes, but the environment shaped those genes. That contractor or the mom, what we would say, different moms, different dads, different lifestyles shape the way those genes are going to change, and personality traits are going to come out. We're going to go through that a little bit more detail. So now that we understand the basics of genes, we're going to go and dive a little bit deeper and think of the variations of genes. So how do you get the traits from your parents? So we're going to look at alleles. And what they are variations of genes. You have two types, homozygous, which are alleles are the same, and heterozygous, which alleles are different. So what are alleles? So I want you to think of eye color. And we think of dominant and recessive. And I don't know if you've seen the chart with the 4 different boxes. But if you think of eye color, well, your mom has blue eyes and your dad has brown eyes. And you had brown. So how did that happen? That's alleles. Your mom is going to carry 2 sets of genes for eyes. She may have 2 sets that are blue. She may have well, she has blue, she has 2 sets of blue. If your dad has brown eyes, he may have a brown and 2 sets of brown. He may have a brown and a blue. So we're going to look at that and explore what are your chances of inheriting that. That's what an allele is. It's the 2 parts of the gene, of the trait that makes up the gene. So if you think of blue eyes, do you have 2 traits that are both brown, or do you have a dominant trait that's brown and a recessive that's blue? So whether or not one receives dominant or recessive alleles from their parents determines a number of characteristics. So we're going to go into this a little bit more, but an example is sickle cell disease occurs when both parents pass on the recessive allele for their disease. So when they're talking about passing on diseases too, that's why they always ask, is it recessive or dominant? Cystic fibrosis is another recessive gene. So neither parent could have it, but they could both be carrying it recessively and pass it on to their child. We're going to go into this a little bit more detail as we lecture through this. So, as I was talking about the blue eyes and brown eyes and dominant and recessive, this picture is going to illustrate that concept. So we want to think of that dominant recessive mode of inheritance. And we're going to use the example of PKU, which is just a form of disease you can carry. So you want to look at the father and the mother. A big N means it's dominant. Anytime you have a capital letter means it's dominant. Anytime you have a lowercase number means it's recessive. So we want to look at this father and mother, and you remember you're going to have 2 sets of genes on it. So he has a big N and a little p. So that means he is a carrier, because he's a carrier. He doesn't have it, he carries it. And then you look at the mom. She has the exact same thing. She has the big N, little p. So whenever you have a dominant with a recessive, it's always going to be dominant. You can have 2 dominants and be dominant, or you can have a dominant and recessive, and you're always going to be dominant. So neither of them have the disease. So they may not even know they carry it, because neither of them have it. So then they have 4 children. And you can see the first combination. These are the 4 different combinations, so they have a 1 in 4 chance of getting it. Each child does. So to have a normal child, you can have a capital N and a capital N. So that means that they have just no trait of that PKU. So they don't carry it. None of their kids can have it. They are completely normal. They cannot pass it on, and they will never have it themselves. Then you see the 2 in the middle are carrier, because they're just like their parents. They have a big N, which is dominant, and a little p that's recessive. And remember, if you have a dominant with a recessive, the dominant you will always have the physiological trait of the dominant. So those are 2 carriers. They don't have it, but they have the gene inside, part of the gene inside, to pass it on. And then the last one is the actual person that has the PKU. Then they have 2 little p, so they have 2 recessive. And the only way you can have it, a recessive trait that be show physiologically or show is if you have two recessives together. So you can see in this diagram that there, out of the three, there's a 25% chance that those two carriers can pass it to their child. So because it's a recessive gene. So this disease is passed recessively. So that's why there's only a 25% chance that their child can get PKU, because to manifest physiological symptoms of a recessive gene, you have to have both recessive genes in it. So this is a table of common phenotypes. So remember, phenotypes is the observable characteristics, be it personality, physical, behavioral. It's the observable qualities that are formed. So it's actually what the gene shows, not what you have. It's actually what the gene shows. So we want to look at some common phenotypes associated with single pairs of genes. That just means that you have one gene affecting it. So I want you to think about the people in your family and think of what you look like. Think of what your mom and dad look like and what you like. Do they have curly hair, and did you get it? Or normal hair, dark hair, normal color vision, or they have normal hearing, their blood type. So let's look at some of these and see how they're passed. So curly hair, so dominant phenotype and recessive phenotype. So what that means, that curly hair is dominant. So when you put a curly hair person and you put a straight hair person together, you're more likely, you're 75% chance of having that curly hair, right? Because curly hair is always dominant. Because think back at that PKU picture. To have straight hair, you have to have both recessive genes. So look at normal hearing and deafness, right? 75% chance. So if you have a parent that has a carrier who's a carrier of deafness, then, or you have a parent that's deaf, you're going to depend on how well you're going to pass that on. But you're always going to, normal hearing is always going to be more dominant than deafness. So you're more likely to have normal hearing, more likely to have normal vision. Type A blood is more dominant. So that means if you're AO, you're going to be, your blood type's A. To be an O blood type, which I am an O blood type, that means that I have both my genes are OO. I can't have AO because A is dominant. It's always going to show the characteristic of the dominant trait. Same thing with B. So your RH factor, your positive is always more dominant over your negative. So if you get your mom's positive and your dad's negative, and you're going to be more likely to get the positive if you have a positive negative combination. So I'm O negative blood, so that means my gene looks like I have two recessives. I have an O, and I'm negative. So my dad, I know, is O negative, and I know my mom is B negative. So I know that my mom, because I have an O, she's a carrier of an O, because that's the only way that I could be O. So her blood type is actually BO, but B is the dominant phenotype, so she's going to show as a B. So just kind of look at your family and think of your characteristics and traits, and you can see if you got a recessive or dominant phenotype, and then you can kind of look and know what your genes your parents have, because to have a recessive, that means your parents have to be a carrier. One of them has to carry that, even if they don't show that trait. Now, if they show that trait, they have two of those genes. If they were recessive, straight hair, they have the two little, say like it's an S, two little S's. So that means that you have a 50% chance of actually getting that trait. So this is a good table to look at, and just think of your family and think of that dominant to recessive. Dominant with a recessive is always going to, the phenotype is always going to show dominant. And to be recessive, you have to have both recessive parts of the gene to show the phenotype as recessive. So now we're just going to review autosomes, sex chromosomes, and sex cells. So when we get to the next lecture in chapter 2, preconception, you're going to understand how that happens. So autosomes are the 22 pairs of chromosomes. So that's all the chromosomes except the sex chromosomes. And the sex chromosomes is the 23rd pair of chromosomes, and it's what determines if we're a male or a female. So a female is two X's, and a male is XY. So that's why they always say that the male determines the sex of the baby, because it's the only one that has an X and a Y. So if you put it together, right, it's always going to determine, because a female can only technically have two X's. It can only pass on its female traits. A male can pass on the X part and the Y part. So they actually determine the sex of the baby. And then gametes are sex cells. They're the sperm and the ovum. So that's what comes together to form the zygote, which is formed when the sperm and the ovum unite. And that is the first part of forming the first cells of conception of life. So that is how our chromosomes come together to form life, to start that process that we've been talking about, the DNA and all that, and entering the cell. And we'll be going over later on in this chapter, how that comes to be in preconception. But I just want you to understand the autosomes are the 22 of your chromosomes. The 23rd pair is the sex, and it has two parts. The gametes is the sperm and the ovum, and then you have the zygote when they form together, and when the sperm and the ovum unite, that forms the zygote, and that's the first part of conception. So now we're going to look at the mechanism of heredity when you have one or extra chromosome in your sperm or egg cell. So development is disturbed when there are more or less than 23 chromosomes in a sperm or egg cell. And Down syndrome is a great example of that. It's caused by an extra 21st chromosome. People with Down syndrome have different characteristics. And you remember that genotypes that we saw earlier. So something on that 21st has an extra chromosome. So you're going to see probably another XXY that'll look like. And because of that, they develop not various differences in physical, psychological, cognitive features. So that makes up a array of different manifestations. Not just one thing is different. The whole way they process, the way they physically develop is different. People with Down syndrome have almond-shaped eyes and folds over the eyelids. Their head, neck, and nose are usually smaller than normal. During the first months of life, development of babies with Down syndrome seems normal, but thereafter their mental and behavioral development begins to lag behind the average child. So for instance, a child with Down syndrome might first sit up without help around 1 year, where a normal child would be 6 months. Walk at 2 years, where a normal child would be 1. And talk at 3 years, which is way, beyond, behind, usually beginning at 18 months for a normal child. They're going to reach many of their developmental milestones months or even years behind children without Down syndrome. So by childhood, most aspects of cognitive and social development are seriously delayed. So what are the odds that a woman will bear a child with Down syndrome? Well, it's increased markedly as she gets older, from about 1 in 1,000 during her 20s to 1 in 50 during her 40s. So you might ask, well, why? So one of the reasons why is that woman's eggs have been in her ovaries, so you're born with all the eggs that you're going to be born with in prenatal, and we'll learn about that. Eggs may deteriorate over time due to aging, and older women may have greater exposure to hazards in the environment that may damage eggs, like an X-ray. So that's why many women are taking the option of freezing their eggs so that their eggs are 25 if they want to have a baby at 40. A lot of different instances where they want to have careers, they're not ready to have families, they can't afford them. Yep. Many reasons why women would delay having children, but they don't want to have 40-year-old eggs. That would increase their chances of having these abnormal chromosomes. So a lot of people are freezing their eggs for that. Abnormal sex chromosomes can also contribute to development, and we're going to look at the table on the next slide to look at abnormal sex chromosomes. So this is an example of an abnormal and the other 23 groups of chromosomes. So if you think of abnormal development or an extra X or Y in the 23 chromosomes, Down syndrome is the example you want to think of. So this is a table of abnormal sex chromosomes that also disrupt development. So you're going to look at the 4 different types, and it tells you what the abnormality is. So you have Klinefelter syndrome, and that is an XXY, so they have an extra X. 1 in 500 male births, so they're going to be a male because they have a Y. The characteristics are tall, small testicles, sterile, below normal intelligence, and they're very passive. The second one is an XYY complement, and they're going to have an extra Y, and that is 1 in 1,000 male births, and they are tall. Some individuals apparently have below normal intelligence. They have more risk for learning problems. The third one is Turner syndrome, and it's lacking, so it's an X, so it's actually a female, right? So it's 1 in 2,500 to 5,000 female births. So you can see this is going in order of how what the chances of having it. So short, limited development of secondary sex characteristics, problems perceiving spatial problems. And Turner syndrome is rare because you can see that, right? 1 in 2,500 to 1 in 5,000 female births. And then the last one is XXX syndrome, 1 in 500 to 1,200 female births. Again, it's a female, right? Because all we have is X. Usually caused by malformation of an egg or a sperm in the embryo development. That's usually why. It's normal. They have normal stature, but delayed motor and language development. And in Turner syndrome, it's usually called Fragile X syndrome as well. And it's, again, delayed speech and language by age 2. So that's just a look at some of they're rare, abnormal sex chromosome distribution if you have them as your development. So let's just take a look at that and understand how having not the right amount of chromosomes can affect development. So now that we understand genes and genetic coding and that blueprint, now we're going to understand how the environment and development influences those genes and heredity. So we're going to talk about twin studies, and we're going to talk about polygenic inheritance. So we're going to ask the question, are traits controlled by a single gene or many? So the example is extroversion. So are you an extrovert or an introvert? So are you outgoing or are you extremely shy? So I want you to think of people in your life, or you or a friend or a family member. Are you outgoing or are you shy? Did you have a parent that's like you? If you're outgoing, are they outgoing? Or if you're shy, are they shy? Is it extreme? Are you super, super outgoing? Are you super, super shy? Or are you somewhere in the middle? So this is an example of a polygenic inheritance because because extroversion and introversion is made up of a multiple of genes that make us those traits. So what it's saying is that if I'm an extrovert, I might have 4 characteristics. I might have a capital A, capital B, capital C, capital D. And if I had all those traits and I was all of that, so I have those 4 genes and I have dominant for all that, I'm going to be a super extrovert, right? I'm going to be like Oprah Winfrey. I'm going to be that super, politician because I'm going to be so extroverted. If I have the recessive part of those 4, like the lowercase a, lowercase b, lowercase c, and lowercase d, I am the most introverted person you know. I am that person that, can't speak in public, sits in the back of the room, can't talk to anybody. That is my extreme. So I don't know about you, but in my life, those 2 things are rare, right? Most people are somewhere in the middle. So that's what they're talking about, polygenic inheritance. It's a range. So I might be more extrovert than introvert. So I probably have maybe a dominant A, a dominant B, but maybe a recessive C and a recessive D. But I'm like half and half. Or if I'm, a little bit more extrovert, I might have 3, like a dominant A, B, and C, and a recessive D, because those 4 genes make up extroversion to introversion. So it's a combination. It's It's a range. So that's why you're not all or nothing. And if you are, those are rare because that is a combination that you have to hit all of those genes. So it's less probability. What they did is they looked at twin studies exploring the issue of heredity, and they looked at and saw how identical twins, so that means they're monozygotic, they've got the same genetic blueprint, the same DNA, and dizygotic are fraternal twins, which, basically it's just having a brother or sister at the same time with you. You don't share that identical gene makeup. You share similar gene makeup because you come from the same parents, but not identical. So what they found, if monozygotic twins, so those are the identical twins, are more alike in characteristics than dizygotic twins, heredity is implicated. So that means it means that genes are causing it. But if not, then that means something else might be causing it. And then they looked at adopted children can be used in studies on heredity. So they asked the question, is it nature versus nurture? Remember what we learned from last chapter. Is it your genes that determine what you are, or does nurture play a factor in it? So let's take a look and start to answer some of those questions. How do genes work together to influence behavior? Well, heredity and environment interact dynamically throughout development. So the example they give in your book is the PKU in infants and the onset of menstruation in girls. So as we talked about earlier, PKU is a homozygous recessive trait. So that means you have to have both recessive genes from your parent to have it. And so what they found with that disease, children are fine until they eat certain foods. Certain foods, the environment triggers those genes. So the genes in themselves are fine. And if they never ate a piece of meat, they would never get the physical characteristics of the gene. But when they start getting the meat, they begin to have that physical characteristics. So the physical characteristics of PKU is in damage to the nervous system, leading to intellectual disabilities. So one of the things they test your baby after it's born is the little test in the heel is PKU. Because if you have it, then they can make sure you never get any of the physical symptoms because they know you have the gene and they can alter the environment. You can go on a special diet that doesn't include meat, certain meats, chicken, eggs, and other things, so that you never trigger that gene. So that's an example of your environment changing your blueprint, because your blueprint says that if I eat this meat, it's going to trigger this gene. But if you never eat it, doesn't mean the gene's not there. The gene is always there. You can always have the potential to get it. It just means that you can change your environment to affect how that gene is worked. So remember, the genes, DNA is just a blueprint. The environment has a lot of influence on how those genes are triggered. It's very complex, not straightforward. Some cancers, you have some genes in your body that trigger, cancers and stuff. So a lot of studies that they've gone to. So that's one way that the environment can affect the genes. Another way is the menstruation in girls, that they found that stress in young girls can actually stimulate that gene that causes menstruation early. So again, that's the environment. If that childhood is not stressful, or what constitutes stress for that child does not happen, she'll have a normal menstruation start time. But if something stressful happens and it triggers the gene to turn on, then that's the environment affecting the gene. The gene is always there. Whether it's turned on is depending on the environment. So that's an example of how environment interacts dynamically through development. So you'll have a totally different childhood, right? So once they figured out how to test for PKU, there are many different, that child life is completely different. You have two children, both have that same gene. One was tested and they found out, and they had a correct diet for him. The other one didn't know, and he ate meat and stuff, and he developed neurological difficulties. Same gene set in their body, but the environment affected how their physiological and development occurred. Genes can also influence the kind of environment to which a person is exposed. So your book gives you the example of how might a teacher treat a bright child differently than a disengaged one. So think about it. you're sitting in the classroom and you have the children that raises their hand, me, answers all the questions to the one that's sitting in the back and not asking. Who is the teacher going to pay more attention to? Who's going to get more one-on-one treatment? Who's going to have their skills fostered and, mentored? The one that's more engaged and is bright and engaged, or is the one that sits in the back and is not? So what this is saying, because of the gene, so she's genetically predisposed to be maybe more outgoing, have a little bit more intelligence. It's just a part of it. But because of that, and because of those personality traits, she's changing her teacher's environment. Her genes are affecting the environment that she's learning. Because of those traits, she's forcing the teacher to pay attention to her, to mentor her more, and to, again, foster that intelligence. So that other child in the back may have that same, underlying intelligence skill, but maybe he is not extroverted, or he doesn't speak up, he doesn't foster that. Because of that, he may have a very different developmental process through his school age years, because he's not being mentored, his genes aren't being fostered, so they may not have. He's not put in the environment to bring out all those. To trigger his gene that for maybe critical thinking, intelligence stuff. So he never develops it, and therefore has difficulty in school, maybe doesn't get in college, because she has the personality traits that foster that gene, she's changed the environment to make that gene be triggered and become successful in that dream. Whereas that other student may have the same potential, but because his other genes together didn't influence the teacher to pay attention and to foster it, his gene was never fully triggered, and so his development was a lot different. So think about that when you're in classes, or if you have kids. How is your kid reacting? How is their personality? If you want to become a teacher, how are you going to engage those non-engaged kids? Because if that child changes the behavior, you don't want to miss out on those kids that have their genes there. They have them, ready to use, but just because their personality or other gene factors that they're not getting that same mentoring, because they are not influencing the environment as opposed to what a more bright and engaged child is. So just some things to think about. I want you to picture your friends now. Are your friends like you? Do they like the same things? Do they have the same personality? Or do you go for the opposite? Well, with niche picking, it's the process of deliberately seeking environments that are compatible with one's genetic makeup. So it's the idea is that like people seek out like people. Children who are outgoing often like to be with other people and deliberately seek them. So because of that, they have the gene for being more outgoing. They seek out people that are more outgoing, so they're going to foster that. So it's that idea, what came first, the chicken or the egg? Were they genetically more outgoing, so they sought out people that were more outgoing, and then in turn they came to be more outgoing? So what started it? Or is it a combination of both? Niche picking is saying that they're genetically more likely to be outgoing, so they're going to gravitate towards those people. And because they do gravitate, then the environment that they're in is going to foster and make that gene really grow. So it's going to have the phenotype of both. The genotype might be there because it's already there, and then the phenotype, the physical characteristics, are really going to show because you sought out the environment that your genes already wanted you to have. Then I want you to think about your family. Do you have brothers and sisters? Did your parents treat you exactly the same? Are you guys were raised in the same house? Did you guys have the same rules? Did you live the same childhood? Are you guys the same person? Well, of course not, right? Even within families, parents aren't the same to the same child, right? I know my sister was 7 years younger than me. She was treated way different than I was. And even my brother, who was 3 years older, even though we lived in the same house, had the same parents, we each had individual childhoods. So environmental influences typically make children within a family different because a parent isn't the same parent they are to your sibling as they are to you. Behavioral genetic studies show that siblings are not much alike in their cognitive and social development. And the reason is just like I said, because parents parent to the individual child, to your strengths and your weakness. We're not all the same, so their styles aren't all the same. And then depending on your context of their life, think again about that historical context. Timing is everything, right? My brother and sister are 10 years apart. My parents are two different people 10 years apart. Their styles are going to be different. It's a different household. So that's what they're saying, is that environmental influences the parents parent differently to the individual child. And because of that, siblings through research have not been alike in their cognitive and social development. So this figure shows the environmental influences typically make children within a family different. The family environment affects each child in a unique way. We talked about that, right? Did your parents parent you the same as your sibling? No, right? Parents influence their children by providing genes and by providing experiences. Children's genes and their environments work together to shape development. So what I want you to remember here is that you have non-shared forces that make children different from each other. Parents may be more affectionate with one child. They may punish another child more. I know my brother, being the oldest, he was punished a lot more than my sister, being the youngest. Totally different environments, came out differently socially attributes. So birth order. So again, like I just said, birth order makes a difference. Greater expectations maybe for the firstborn than for the lastborn. Maybe they're just tired. Maybe they're more mature. Parents change as well as the child. So it makes a difference. And in turn, it makes your siblings different. So even though it looks like the same environment, it's really not. It's a unique, multiple environment. It's a unique for parent to child 1, parent to child 2, and parent to child 3. 3 unique environments. They're non-shared forces. So they have, and then they have a different gene makeup. So when you think about, you have 2 siblings, right? One of them may inherited the extrovert, and one may have maybe got that recessive introvert. So you're going to be doing 2 different parenting styles. I'm going to parent the introvert differently than the extrovert, right? Maybe, like I said, that introvert I'm not with them as much, or I leave them more alone, or I have to force them to do things where the extroverts are always there. Maybe they get more attention because they're just always with me and they want to do things. So they have different niches, different people, but they're the same family. So genes influence your environment, your parents' genes influence the environment, and the historical time, the birth order when you were born influences. So heredity, environment, development, genes, all that makes up you. So we have the blueprint in our DNA. We have the environment that triggers the DNAs to come to life. And then we have development, which is all that in between. So part of it's nature, part of it's nurture, but all of it together is you. So we're going to come and carry this on in the next chapter. And we're going to learn about preconception to birth. See you guys then.